Hello and good evening to CDT 23 committee members and all participants. My name is Fariza Aina and I am from University Putra Malaysia. Currently, I am pursuing my PhD under supervision of Prof. Dr. Nur Azhar. So today, I will present my research project entitled Aptamer Conjugated Chitosan-Based Nanocarriers for Possible Targeted Diagnostics of Non-Muscle Invasive Bladder Cancer, or NMIBC. So first thing, why do we concern about bladder cancer? So let's have a look on global epidemiology of bladder cancer. Based on data provided by World Health Organization or WHO, bladder cancer has been listed as 10 most deadliest cancer that cause mortality worldwide. Based on continents, Asia has become on the first place with the highest case in Singapore, followed by Thailand and Malaysia on the third place. Generally, bladder cancer can be classified into two major categories, which are NMIBC and MIBC. In this current research, I will focus on non-muscle invasive bladder cancer, NMIBC, as it constitutes 3/4 or 75% of the incident cases, and the rest is MIBC, which is muscle invasive bladder cancer. So let's have a look on pathophysiological invasion of bladder cancer. For NMIBC, the invasion of cancer involves three stages, which are TIS, urothelial carcinoma in situ, TA, non-invasive papillary urothelial carcinoma, and the last one is T1, invasive into lamina propria. So, the first line for diagnosis and treatment for NMIBC is transurethral resection of bladder cancer tumor, also known as TURBT. This procedure involves the biopsy of tumor and resection of tumor from the bladder. So, to prevent the reoccurrence of cancer, intravesical chemotherapy is needed. Somehow, this treatment has several limitations such as non-specific biodistribution, insufficient drug concentration at lesion site, low therapeutic indices, as well as intolerable cytotoxicity. Hence, targeted drug delivery system, or we call as TDDS, has been developed to counter all the limitations with their special features, specific biodistribution, sufficient drug concentration, high therapeutic indices, as well as low cytotoxicity. The main objectives in this study is to develop chitosan-based drug nanocarriers conjugated with aptamer for targeted drug delivery system in NMIBC. To achieve these objectives, we do have detailed experimental design based on these four objectives in our next slide. So, these are our experimental design based on previous objectives. First, we synthesize and characterize the nanocarrier, and then next we study the encapsulation efficiency followed by drug release sustainability using UV spectroscopy. After that, we do computational analysis on conjugation of aptamer and the VEGFR1 protein biomarker, followed by experimental analysis using enzyme-linked oligonucleotide assay, or ELONA. And finally, we do fluorescence spectroscopy to evaluate the analytical performance. Next, we go to morphological analysis of nanocarriers. Basically, we have three types of nanocarriers here. In drug delivery, particle size below 10 nanometer is prone to undergo kidney filtration, and particle size larger than 200 nanometer cannot pass through the epithelial gap. So in this study, particle size between 80 to 100 nanometer is the ideal particle size. As we can see in table 2, based on zeta potential value, all nanocarriers exhibit positive charge, which can electrostatically interact with an anionic mucosal membrane layer of cancer cells. For all nanocarriers, the encapsulation efficiency obtained is quite high, about 80 to 85%, which is in a good agreement with previous study. Drug release sustainability, the nanocarrier tends to release drug in acidic medium, which mimicking the cancer microenvironment. For all nanocarriers, there is no significant difference in drug release as they exhibit the same formulation except for the additional encapsulation of fluorescent probe purposely for bioimaging. Okay. The conjugation of aptamer biomarkers was evaluated using molecular docking. In this study, as we can see from figure 5, VEGFR biomarker has 6 binding pockets as highlighted in green color. In this study, 2 sequences of aptamer were selected and labeled as aptamer 1 and aptamer 2. For aptamer 1, the binding to biomarker at 2 binding sites, and for aptamer 2, they bind at VEGFR1 biomarker at 6 binding sites. Six, this is the binding of aptamer 1 with VEGFR protein receptor at 2 binding sites. And then figure 7, this is the binding of aptamer 2 with VEGFR protein receptor at 5 different binding sites. Next, we further experimental analysis of both aptamer with VEGFR biomarkers. From the results, aptamer 1 has dissociation constants at 342 nanometers. Meanwhile, for aptamer 2, the KD is 375 nanometers. KD represents the degree of dissociation of complex into free aptamer and free target. High KD value means that they have poor affinity towards the target and vice versa. So in this study, aptamer 2 have high affinity towards VEGFR biomarkers, and the result is tally with computational analysis that has been carried out previously. Finally, the evaluation of analytical performance of developed drug delivery system using fluorescence spectroscopy has been carried out. Generally, we can see from figure 9 both fluorescent peaks of MN, ZNS and CDS were quenched after the conjugation of aptamer. Next, after the formation of target receptor complex with VEGFR1 biomarker, further attenuation were observed. This is due to the formation of donor-acceptor complexes during target binding. Here is the schematic representation of our drug delivery system. So first, the encapsulation of mitomycin C drugs into chitosan-based nanocarrier, followed by conjugation of aptamer on the surface of the nanocarrier. Then, aptamer conjugated drug delivery system, or we call as DDS, will bind to the protein receptor, VEGFR1 protein receptor or biomarkers, and then promote cellular internalization through endocytosis process. Endocytosis is responsible to facilitate the efficient distribution of drugs towards cancer cell at lesion site. Mechanically, the drug will be released inside the cancer cells to induce killing and suppress the cancer cells. In conclusion, targeted drug delivery system, or TDDS, is an innovative solution for cancer diagnostics with low toxicity, specific biodistribution, high therapeutic indices, and sufficient drug concentration at the lesion site. So these are some publications. So these are the references. For inquiries, you can reach out to us at this email. And thank you for listening.